Hey guys, this is James, Wesley, and John Larochelle here. We are presenting on penicillin for you today. We're going to go through some of the biosynthetic pathways of penicillin and its medical relevance, and also some of the mechanisms of penicillin resistance. Alright, so I'll take us off with the relevance of penicillin. It was first discovered by Alexander Fleming in 1928. So the story goes that Fleming returned to his lab. He was working in a microbiology lab. He was a microbiologist, and he discovered that the plates that he was working on were infected with this fungus. And at first he was upset because the fungus had seemingly destroyed his bacteria, but then he began to wonder why the fungus killed his bacteria, and he began isolating compounds from the fungus and came across penicillin and its ability to kill bacteria. But he wasn't much of a chemist himself, so it took the work of Howard Florey, Earthchain, and others at Oxford University, who were chemists who were able to isolate the active penicillin compound and actually take full advantage of it and use it for medicinal purposes. And its discovery of penicillin is credited with being one of the most important discoveries of the 20th century. For one thing, it's estimated to have saved over 200 million lives, this being from mostly wars and other just normal infections where if someone received some sort of injury, an infection would lead to their death, almost certainly. And so before penicillin, there was widely used were sulfonamides for being used against bacterial infection, and these were effective, but definitely not as effective as penicillin. Penicillin is an actual cure for bacterial infection because it kills off the bacteria. And so, yeah, penicillin, it does not act on any human enzyme, and thus it has a very low toxicity. And our sources for this slide come from this article on what if Fleming had not discovered penicillin, and from the Centers for Disease Control and Prevention. And a little bit more on the relevance of penicillin. It was actually awarded, the discoverers of penicillin were awarded the Nobel Prize in Physiology or Medicine in 1945 for their discovery.
So a little bit about the structure of penicillin here. We see that it contains this thiazolidine ring in red, which is this 5-membered ring, which is fused to the beta-lactam ring in blue, which will turn out to be a critically important portion of the molecule via a peptide linkage. Additionally, penicillin is in the class of molecules that belong to the family of beta-lactams because of this 4-membered ring here. So penicillin G is the most active compound, which is isolated from the Penicillium chrysogenum fungus. And so this compound is what is actually used for antibiotics in industry, which is different from the penicillin that Fleming and his colleagues originally discovered, but penicillin G was determined to have the most antibiotic properties. And our sources for this come from the article Antimicrobial Agents, Antibacterials, and Antifungals from Brixier and his companions.
All right, so I'm going to walk you through the biosynthetic pathway of penicillin production. It's a fairly complicated mechanism. Some of these enzymes do some crazy work, but we'll only touch on 2 of them in relative depth, and the other one is too complicated to try and cover. And for this information, we're going to be drawing from Brackage and his group, who wrote a paper on the biosynthesis of penicillin, and he published in Advances in Biochemical Engineering. There's 3 main enzymes that are involved. The first was ACV synthetase, which is conveniently named because of the 3 compounds that it uses to synthesize the intermediate. It uses alpha amino adipic acid, a cysteine, and a valine, and thus it's ACV synthetase. The second enzyme is isopenicillin N synthase, and the third is acyl-CoA, isopenicillin, and acyltransferase. The first enzyme here, again, it takes those 3 substrate molecules and it forms this complicated molecule here. And you can see that it's close to being the final penicillin product. The thing that's missing though is the formation of the beta-lactam ring here and the formation of this 5-membered ring here. And so what's going to need to happen is we're going to have to lose these protons here, and a proton here, and here. Oh, sorry, and here. And I'll show that again. Yes, so this is the second enzyme, also penicillin N synthase, which, like I said, is going to get rid of 4 protons to form the beta-lactam ring and the 5-membered ring shown at the bottom to form isopenicillin N. And this one we'll cover in a little more depth. So it involves a non-heme iron 2 dependent oxidase, and the enzyme catalyzes the removal of 4 hydrogen equivalents to form those 2 rings that I was talking about. And the reaction requires a ferrous iron, molecular oxygen as a co-substrate, and ascorbic acid as an electron donor. This is again a little bit more about the enzyme. Again, it needs to form those 2 rings that are shown in red and the 2 substrates, oxygen and ascorbate, and it's producing 2 waters. And highlighted at the bottom are the 4 hydrogens that need to be removed, or the 4 hydrogens that were removed in the isopenicillin N product. And again, the mechanism is fairly complicated. It first starts with the deprotonation of that sulfur up top, and that goes to form a bond with the iron, and you lose that hydrogen obviously to the base. And then oxygen comes in here, and it's actually in the form of superoxide, and it does single electron chemistry as shown by all these arrows here. You're moving single electrons all over the place until you form this compound here, where now you're doing, let's see, it's a hydrolysis where you're removing water, and the double bond from sulfur is going to attack the iron and form a covalent link with that. And you're also, the reaction here leads to the formation of an iron 4 up here, and then that again allows for some single electron chemistry to happen, and the 4th and final hydrogen is able to be removed here, and you are left with the formation of the beta-lactam ring and the 5-membered sulfur nitrogen ring here. And the third enzyme in this pathway is acyl-CoA, isopenicillin N acyltransferase. This mechanism involves the addition of phenylacetyl-CoA, and basically the phenylacetyl-CoA adds the acetyl and phenyl group to this area here, this R area, and what leaves is the alpha amino adipic acid that was involved in the first enzyme, where we took the alpha amino adipic acid, combined it with a cysteine and a valine, and got our isopenicillin N shown here. And yes, so this leads to the final formation of penicillin G, as we saw earlier with this phenyl ring and the acetyl group added on.
All right, thank you, Jonathan. So now we're going to move into some more exciting stuff, the regulation of penicillin biosynthesis. So we're going to start with the carbon source regulation. So glucose versus lactose. Scientists have used glucose in penicillin production, and what they found is that when there's excess glucose in the growth medium for the fungus, this results in reduced penicillin production. So glucose represses some enzymes of the penicillin biosynthetic pathway. The fungus P. chrysogenum that we've been discussing is a strain of fungus that's used for penicillin production that forms penicillin G. This fungus grows when glucose is the carbon source. However, it does not result in the large penicillin production. So lactose, on the other hand, as the carbon source, shows great penicillin production. So therefore, in the synthesis of industrial penicillin, scientists will actually feed both lactose and glucose into the growth medium, so that you can see a result of a growth of the fungi, and then after the glucose is depleted from the growth of the fungi, the mycelia that are formed, and I'll get to mycelia in a future slide describing what it is, those will begin to produce the antibiotic penicillin with the lactose that is still present in the growth medium. And so there are penicillin biosynthetic genes, ACVA and IPNA. And the expression of these genes was shown to be repressed by glucose. So there was evidence using a deletion analysis and experiment that there's a region in the ACA gene promoter region that is actually responsible for the repression of these genes by glucose. And we got this information from the paper Regulation of Penicillin Biosynthesis in Filamentous Fungi and from this Science Direct article. Moving on with some more regulation, we see some regulation by nitrogen. So, in the Chrysogenum fungi, the penicillin biosynthetic genes that I just discussed, IPNA and ACVA, were refused with a reporter gene in a strain of E. coli bacteria, and they refused with that to see the expression of those genes. The scientists discovered that the expression of both of those genes in E. coli was repressed when 40 millimoles per liter of ammonium chloride was added to the growth medium. Therefore, nitrogen serves as a regulator of the biosynthesis of penicillin by repressing it. Furthermore, we see regulation by amino acids. So on the right here in this figure, we see the biosynthetic pathway that Jonathan just walked us through. In this section here, we see the 3 amino acid intermediates that will combine to form this molecule shown here. And so we see that they are downstream to lysine in the pathway. So what happens is that lysine will get degraded and form this amino adipate amino acid, and then it will combine with the cysteine and valine to form this molecule. So scientists discovered that lysine actually inhibits penicillin production, and this operates by inhibiting the enzymes of its own biosynthetic pathway through negative feedback inhibition. So as shown in the picture, if lysine is not able to produce amino adipate, then this amino adipate will not get added with cysteine and valine, and penicillin will not be able to be produced. So they saw that when you added lysine in, it acts according to the principles of Le Chatelier's principle and acts as a negative feedback on the lysine biosynthesis. And the last slide for regulation is a slide about oxygen. So oxygen is also necessary for penicillin biosynthesis because it helps the enzymes in the pathway to function correctly. One of these such enzymes is the IPNS enzyme, the isopenicillin N synthase, which Jonathan has covered. This enzyme is responsible for converting the ACV intermediate to isopenicillin N. And so I'm going to discuss a little bit about those mycelia that I mentioned earlier. So the mycelia and the fungi are it's the vegetative portion of fungi, which are tubular branched filaments that cluster together. And so these filaments are where the isopenicillin N synthase is located. Now, these mycelia need to be aerated with oxygen in order for the synthesis of beta-lactam rings to occur. Now, the synthesis of those beta-lactam rings occur through this enzyme, isopenicillin N synthase. So therefore, oxygen is necessary for the formation of those critically important beta-lactam rings, and we'll discuss why they're so important in the slides to come.
All right, thank you, James. Now we're going to talk about mechanism of action of penicillin. So penicillin acts on peptidoglycan synthesis, and in that way it leads to the cell death of the bacteria because it leads to cell lysis, or it leads to lysis of peptidoglycan. And penicillin is able to penetrate the bacterial cell wall and thus act on peptidoglycan synthesis. And its main way of activation is forming a covalent link to the enzyme transpeptidase, which is involved in catalyzing the final steps of cell wall biosynthesis, the cross-linking of peptidoglycan. So if you remember in our chapter on where we covered peptidoglycan synthesis, we talked about NAM and NAG, and I believe it's the cross-linking of the NAM groups that transpeptidase catalyzes that reaction. And so when penicillin is added, you are forming this covalent linkage here, which becomes this covalent linkage, to the enzyme transpeptidase, which inactivates it, and in that way, no more peptidoglycan can be synthesized, and the bacteria lyses and die. And we're going to cover resistance to penicillin briefly, and this penicillin resistance has been around basically since the beginning of penicillin. It was first discovered in certain strains of Staphylococcus aureus in London, England, when penicillin was first being used. And this resistance is due to plasmid-mediated transfer of a plasmid, which contains penicillinase. And penicillinase is what breaks down penicillin and leads to its inactivation in certain strains of bacteria. And this is all from the source Breskier, hard name to pronounce, and it's his paper on antimicrobial agents, antibacterials, and antifungals. And just a little bit about the mechanism of penicillin. It's just a hydrolysis reaction where you're breaking that peptide bond between the carbonyl and the nitrogen through a hydrolysis reaction. Oh, look at, James is drawing it out. Look at that. That's great. Yep, simple mechanism, but that's all there is to penicillin resistance. And so that's all there is to our presentation as well. We'd like to thank you guys for listening in, and we look forward to listening to the rest of your presentations as well.